Welcome to this session presented by Mrs. Prudence Amito. We are going to be looking at neonatal nursing, and when we talk of neonatal nursing, you cannot escape looking at how the fetus came about or how the newborn came about. So today we shall be looking at some few details of how the newborn becomes a newborn. So let's look at fetal development, and when we look at this fetal development, it's a process that begins right from fertilization. So you're welcome, and I invite you to be attentive as we listen to this presentation. So our first slide is talking of introduction. So when you talk about fetal development, basically we want to look at human life begins as a single cell, which is a zygote, or a singleton, which is a zygote, and the other name of a zygote is a fertilized ovum, which usually comes as a result of sexual intercourse or intercourse. As two people meet together, then the result is always the zygote. So the cells reproduce continuously to form a human body. So after the zygote has been formed, it undergoes a series of cell divisions for the human body to form. So reproduction occurs via mitosis, which is for growth and repair, and then the meiosis, which is basically for gametes formation. So let's look at what mitosis is. So mitosis is a process of producing exact copies of cells. So when we hear about mitosis, basically we are looking at how cells multiply with exact copies. So we have that multiplication resulting in two identical data cells with 46 chromosomes, what we call the diploid cell. So this is actually what happens when the sperm meets the egg. It gives us the 46 chromosomes. Then we have it's also responsible for growth and repair of tissues, as I mentioned before. So what we wanted to know also about meiosis. So meiosis is a kind of a specialized cell division which takes place basically in the testes and the ovaries. So this produces gametes like the sperm and the ova with 23 chromosomes, that is the haploid cells. So meiosis usually takes place after mitosis has taken place. So basically, as fertilization takes place, there is excess number of chromosomes which are produced, and it is via meiosis that's when the number of cells or the chromosomes are reduced to the one which is supposed to be normal. So in most cases, meiosis is division of cells by 4. So there are two steps, two-step process that ensures genetical variations. That is the meiosis and then mitosis. So let's look at gametogenesis. So when we hear about gametogenesis, just a process through which cells are the cell division by meiosis and then mitosis. So basically, it helps us the information of gametes or these other, how do we call it, the reproductive cells. So one of the, like how it begins about or how it starts. So when you talk of gametogenesis, two things should come into your mind. One, that is spermatogenesis, and then what we call oogenesis. So first of which we have spermatogenesis, which is the formation of sperms in the testes. So the formation of sperm in the testes results in 4 spermatozoa per spermatocytes. So each has a head, which is the nucleus, and the acrosomes. And then we have the middle piece and then the tail. So usually what happens during fertilization is that the head, which contains the nucleus and the acrosome, is the one which is very important. Then the middle piece and then the tail. Then we look at oogenesis, which is the formation of ova in the ovaries. So whole ovaries are present at birth. For every female child who is born, is produced with all the eggs. The ovaries are those eggs. So they are all present at birth. Meiosis begins before birth, pauses until puberty, and then when the child reaches puberty, it continues. That's when the egg will begin to be released one at a time, let me say once in a month, and that is a process where we call ovulation, the process of egg being released from the ovaries. So it produces one ovum and 3 polar bodies, which usually degenerates after the egg has been produced. So after that, we have after the egg has been produced, we know that what should take place, or what was tentatively supposed to take place, is either fertilization, or if fertilization does not take place, that means the whole process will be terminated in form of menstruation. So let's look at overview of fertilization. So fertilization has a few points which are very important, one of which is fusion of sperm and ovum to form a zygote. So there must be meeting of these two gametes, that is the sperm and the ova. So when they meet, they give us 46 chromosomes. Then there is the zygote is genetically unique. So after that, there is determination of sex at fertilization, and then we have, how do you know this is female, how do you know the other one is male? Females are always represented by XX, and the males are always represented by XY. So it is determined by the sperm. So if it is the Y sperm cell that has fertilized the egg, then it becomes FX, because we know that the females, they carry XX, and the males, they carry XY. So if the X of the male is the one that went and met with the one for the egg, it becomes XX. That means the child which is going to be born will be female. But if it happens that the Y of the sperm is the one that met with the X of the female, then it becomes XY, and we find that it is a baby boy. So let's look at a few things that prepares a person for fertilization. So we know that we have already mentioned that what happens is from puberty, now the female child will begin to release one ova or one egg at a time with 3 polar bodies. So what happens is this egg, which is released each month, it has a lifespan of about 12 to 24 hours for it to die off or to begin to degenerate. So the ovum has a lifespan of 12 to 24 hours after ovulation. So after the time that the ova or the egg has been released from the ovaries, it stays for about 24 hours before it begins to degenerate. Then we have the sperm lifespan up to 72 hours. So there's a reason as to why the sperm lasts more longer than the egg. We shall look at it when we are looking at details of fertilization. So the most fertile hours of the sperm is also within 24 hours. Then we have some hormones which are very, very key during this process of fertilization, or which is the estrogen. So the estrogen enhance peristaltic movement of the within the fallopian tube, especially if the egg has just been released. Then we have the same estrogen making sure that there is a thin cervical mucus which is produced to make sure that the sperm moves freely without being blocked. It also helps to reduce friction and also reduce the acidity of the cervix and the vagina, to make sure that the sperm moves and is not stuck within along the pathways as it goes towards the fallopian tube. So these are the three things that we should put in our minds if we are beginning to look at fertilization. We look at the lifespan of the egg, the lifespan of the ova, and of the sperm, and then the hormone, which is very, very important, which is estrogen. Now let's look at the process of fertilization. So the process of fertilization begins right from ovulation, actually. So from the time the egg has been released, it moves with that peristaltic movement towards the fallopian tube. Specifically, it comes and stays where we call the ampulla, where fertilization usually takes place. So the egg will wait for the sperm at the ampulla, and then the estrogen within the estrogen hormone will therefore make the cervix to produce cervical mucus, which will enhance movement of the sperm. That is if intercourse, if somebody meets with somebody within that specified period of time. So during each ejaculation, let's say each and every time a man releases, they release about 200 to 500 million sperms. And out of these 200 to 500 million sperm, they are ejaculated within the posterior wall of the vagina. Out of this, about 300 reaches the cervical rim. So what happens is, out of all these 300 million sperms which reaches the cervix, some of which dies on the way as it proceeds to go to the uterus, and also until when it reaches the fallopian tube. So only 100 reach the fallopian tube out of 5 million which has been released just in one ejaculation. So as the sperm struggles to reach the tube, we have what is called capacitation, which takes place, that is sperm maturation. So this capacitation is the removal of the cell membranes of the sperm. There is a cell membrane which covers the head of the sperm. So it's the removal of the cell membrane of the sperm which takes place within the uterine cavity or within the uterus. So capacitation takes place within 72 hours. In the 72 hours, or within the 24 hours, 7 hours, not 72 hours. So within 7 hours, it still remains within the uterine cavity for it to mature. If the sperm was not mature enough, it will stay for capacitation and removal of cell membrane, for its tail to be strong enough, and all those things. So then there is another reaction that takes place, which is there is a reaction called a chromosome reaction, where the sperm enzymes breaks the corona radiator and the zona pellucida. So the corona radiator is the outermost shell of the egg. So the egg releases the sperm releases some enzymes that will allow the egg to be permeable or easily penetrated. So after it has undergone the maturation, the egg becomes strong enough to to go and penetrate the sperm, because what we call the corona radiator of the egg or of the ova is like a gate that is obstructing and taking care of all the contents within the ova. So the sperm releases some enzyme that makes the zona pellucida and the corona radiator permeable. Then the egg also releases some lysosomes or some enzymes to make sure that the moment the first sperm penetrates or enters to fertilize the ova, there should be no other sperm, or the moment the fastest sperm enters the egg, there should be no other sperm entering. So the egg releases lysosomes or enzymes that make sure that there are no other sperms entering after the fastest sperm has entered. And after that, when the two nuclei meet, that is the female nuclei and the male nuclei, that's when we say fertilization has taken place. And when fertilization takes place, second mitotic division, it becomes complete, and then we shall have formation of the diploid zygote with 23 chromosomes from each partners, giving us 46 chromosomes. So from there, the zygote is formed, and after the zygote is formed, then we can see that there would be continuous cell division within the egg or within the fertilized ovum. So that is the process, just in brief, how fertilization takes place. So there are a series of enzymes and reactions and actions which takes place during the process of fertilization. So now we have the zygote, which is already formed. Now we want to know what really happens after the egg has already been fertilized. So this brings us to we have about 3 different stages of after fertilization. So the first stage is what we call pre-embryonic stage or period. Then we have what we call embryonic period, and then we have what is called the fetal period. So from day 1 up to day 14, we call it embryonic period. Then from the 14th day up to 8 weeks, we call it embryonic period. Then from 8 weeks up to term, that is what we call fetal period. So in most cases, there are a lot of cell divisions and multiplication that occurs within the first 1 to 14 days. So let's look at what happens in pre-embryonic period from day 1, or from the first or from 0 hour of fertilization to 14 days following fertilization. So what takes place is it includes multiplication, implantation, and differentiations. So the zygote undergoes cleavage or rapid mitotic division. This is division by, I believe you know. Then there will be formation of morula. So the rapid mitotic division, that is division by 2. So the cells inside the fertilized ovum will continue to differentiate or to divide itself. There will be that rapid division that takes place within the cell. But as that division is taking place, the size of the fertilized ovum does not increase. Remember, fertilization has taken place in the fallopian tube, where at the ampulla. So if this cell is to increase in its size, if it is to increase in size, there is what we are going to call ectopic pregnancy, because the cell will be stuck, or the fertilized ovum will be stuck within the fallopian tube, that it cannot move to come to to the uterus to be implanted. So there will be rapid division of cells inside the fertilized ovum without increase in size. So the cells divide by the third day. By the third day, we already have about 16 cells inside, or what we are calling the morula. So the morula is a solid ball of cells. The solid ball of cells inside the morula, or what we are calling the morula, are cells which have divided inside, and there are about 16 of them. Remember, it started by 2. So this 2, it continues to divide like that. By third day, we have a ball of cell which is called morula, containing 16 cells. So by the 5th day, we are having about 58 cells which are formed, and we called it a blastocyst. So by the time a blastocyst is formed, there is already, let's follow from this slide. From fertilization, we are having just one egg has been released during ovulation. Then when the egg was fertilized, we have zygote being formed, and we are having 2 cells inside, as you can see, the red and the blue one. Then these 2 cells will begin to multiply. As you see, stage 4, 4-cell stage. Then we have the morula being formed. At this morula, we are having 16 cells already. So from 2, it kept multiplying by 2, by 2, by 2, until when it reached 16. If the number reaches 16, we shall call it a morula. Then it will continue to multiply from inside like that until when it reaches 58. If it reaches 58 cells, we call it a blastocyst. By the time the cell is reaching 58 inside, it has already moved from the ampulla up to the uterus, or the uterine cavity. Or by the 5th day, it has already come into contact with the uterine cavity. So this is what we are calling a blastocyst. It has a outer cell mass, which we call a trophoblast, and it has a inner cell mass, which we call a embryoblast. So the embryoblast becomes the embryo, it becomes the yolk sac, it becomes umbilical cord. Then the trophoblast becomes the placenta and the chorion. Remember, you can see it is the outer part. So the outer part is the one that becomes the placenta and the chorion. The inner part becomes the embryo, becomes umbilical cord, it becomes the amnion. Are we together? So in this same blastocyst, we are saying that there is something called a blastocell. So a blastocell is a cavity which is formed and is filled with fluid. Okay? So that is still happening within day 1 to day 5 or day 7. So implantation occurs from 6 to 10 days after fertilization. Remember, we said the blastocyst reached the uterine cavity by 5th day. So meaning by day 6, if the fertilized ovum has already come into contact with the uterine cavity, implantation can begin to take place. So the blastocyst implants in the upper posterior part of the uterine wall. That is what we call the fundus. So the trophoblast implants invades the endometrium and form the chorionic villi, which is a finger-like projection which is invading the fundus. So endometrium, immediately when the fertilized ovum reaches it, its name changes to what we call decidua. So this decidua has about 3 layers, or the endometrium at that time will have 3 layers. One of which is decidua, now we call it decidua capsularis, which covers the embryo. Then we have decidua basalis, which is beneath the embryo and is the one that forms the placenta. Then we have decidua vera. Some people call it this paritalis. So for it is the one that lines the rest of the uterine wall, which later also joins with the decidua basalis, but in later pregnancy, later weeks of pregnancy. So that is what happens from 6 to 10 days following fertilization. So that is how implantation takes place. Then from day 10 to day 14, we have formation of germ cells or germ cell layers of the blastocyst. We still remember the blastocyst is that cell which has 58 is a ball which has 58 cells inside. So the blastocyst has the ectoderm, which forms the skin, the CNS, the eyes, the glands, and the hair. Then we have the mesoderm, which forms the muscles, the bones, the heart, the kidney. We have the endoderm, which is the innermost layer. It forms the linings of the GIT. Let me say like all the mucosas is formed by the endoderm. Respiratory tract, the liver, the pancreas, any lining of the oleotube will be found at there, including connective tissues. The connective tissues are found in the mesoderm. So there is formation of these germ cell layers from day 10 to day 14. That is cellular differentiation. Then we have embryonic membranes, which are also formed. We are still in pre-embryonic period. So one of the membranes which are formed is the chorion. So the chorion develops from the trophoblast, if we remember. Let me take you back. That is the trophoblast, which is the outermost part. So the chorion will form from the trophoblast. And remember, the chorion is part of that finger-like projection which entered the endometrium. Then we have, it contains chorionic villi for placental development. So there will be that finger-like projection to go and reach out to maternal veins, which will be in position to absorb nutrients from the maternal blood pool. Then we are having amnion. Amnion develops from the ectoderm. So it forms amniotic sac that surrounds the embryo. For it comes from the inner cell mass of the blastocyst. So our next is amniotic fluid. So we need to know where this amniotic fluid comes from. So the amniotic fluid comes from the inner cell mass of the blastocyst. Inner cell mass of the blastocyst. So the inner cell mass of the blastocyst, there will be a cavity which is created. And when the cavity is created, we shall have a few cells, ions, which are permeable to this blastocyst and the skin and the fetal skin. So cells like sodium and chloride are permeable to through the fetal skin and the maternal plasma. So these are the things that accumulate in the blastocysts or blastocyst cavity, or the other blastocell that we talked about. Let me show you this cavity. As you're able to see this cavity, the blastocyst cavity. So this cavity is formed so that there would be that filling of fluid inside, and this fluid at this early stage will still be isotonic. Okay, it will still be isotonic. As the pregnancy continues to grow, at some point it will be filled, the amniotic fluid will contain fetal membranes, some maybe fetal urine, and some secretions from the lung and all those things. But in the early stages, the amniotic fluid comes from maternal plasma. Okay, the maternal plasma and fetal skin. So the fetal skin will have some solids. There's sodium and chloride which penetrate the fetal skin to come and which penetrates the fetal skin. So when that happens, remember where there is sodium, water always follows. So that is how amniotic fluid comes into play. So this amniotic fluid forms because it is like a cushion, like a bed for the newborn or for the neonate or for the fetus or for the embryo at that time. So it maintains temperature for the neonate or for the fetus. It supports its growth. It prevents adherence to the amnion, and also it allows free movement. That's why when you're doing palpation, you're able to feel that the fetus can actually move around and all those things. So the volume of amniotic fluid, the normal amount of volume of amniotic fluid is 700 to 1000 milliliters. Then if somebody is having less than 500 milliliters, we call it oligohydramnios. Then if somebody is having more than 1500 milliliters, we call it polyhydramnios. So that is briefly what you need to know about amniotic fluid. Then we have something called the yolk sac. So the yolk sac is also formed from the inner cell mass of the blastocysts. As we are able to see, it is inside, it's not outside. So it forms from the inner cell mass of the blastocyst, which we call the embryoblast. So for its work is to provide nutrients to the embryo in the early stages. Remember, the placenta is not yet there. So where does the embryo gets its nutrient from in the first 6 weeks? From the yolk sac. So later, as the placenta forms, the yolk sac becomes part of the umbilical cord. So it provides primitive red blood cells for the first 6 weeks, just to make sure that the fetus receives some kind of nutrients. So we are seeing amniotic fluid is beginning to form. We are having the amnion, we have the chorion, albumin, yolk sac. So what you need to know is, in most cases, when you take a mother 6 weeks for scan, what you see even before you see the embryo is the yolk sac. So the presence of the yolk sac alone is able for us to make a diagnosis that somebody is actually pregnant. So we have the umbilical cord, which is formed from the amnion. So the umbilical cord forms from the amnion, and in most cases it contains one vein and 2 arteries. So the one vein carries oxygenated blood, and the 2 arteries carries the oxygenated blood. So this umbilical cord in most cases is covered with what we call Watson jelly. So it connects the embryo to the placenta via chorionic villi. Then now we have formation of what should I say? We have these issues of twins and all those things. How does it come about? So one of which we have fraternal or dizygotic. This is where we have 2 ovaries and 2 sperms meeting. That means at some point, especially for people who take those hormonal drugs for ovulation to take place, there might be possibility of you releasing more than 1 egg in a month. If that happens, yes, there is possibility of you carrying twin pregnancy. Okay, so that means 1 sperm will fertilize 1 egg, and the other sperm will fertilize the other egg. So we have 2 ovaries and 2 sperm. When that happens, there is possibility of having 2 placentas, 2 amnions, 2 chorions, and all those things. So more common with increased maternal age and parity. Apart from those who are taking those hormonal drugs for ovulation. It can also be because of increased maternal age and parity. That's when you will find that even if you don't have any history of twin pregnancy in your or multiple pregnancy in your home, there's just that you carrying multiple pregnancy. Then we have identical or monozygotic twins. So when we look at monozygotic twins, we are having one zygote which split. So it was one egg and one sperm, but due to maybe rapid cell division, it is splitted and they became two. So when they became two, it is one sperm and one egg that splitted itself. So the structure depends on the timing of this division. If during the cell division, maybe the zona pelvis, the corona radiator was not hard enough, then the egg got splitted. So that is how twin pregnancy is able to come about, or how fertilization takes place. So that is basically how fertilization takes place and how a zygote is formed and what follows it after it has formed. So thank you so much for listening.